engage with the world around us. It showed us that we didn't know nearly as much about the world as we thought that we knew. So Hooke used his microscope to look at thin slices of a cork tree, of the interior of a cork tree. And without magnification, when you look at a cork tree, you can look at its bark, which I read descriptions as being knobby or toothy, or you can look at the leaves, which are green and are kind of conversely described as both waxy and shiny. You can cut down the tree if you're so daring and look at the concentric golden and reddish circles that make up the heartwood, or the swirling lines of the cambium. But until the microscope was invented, you couldn't really see anything more than that. So in 1665, Hooke used his microscope to look at just so many things, and he published what he saw in a manuscript that was called Micrographia. And one of the things that he looked at, as I said, was this thin slice of cork. And he introduced the world to the concept of a cell, a small repeating component of a larger biological structure. And he made us understand that things that we see with the naked eye are so much more complicated. And we continue to use microscopes to realize that everything on this planet is so much more interesting than what we can initially see. We realize that every organism is composed of cells, and those cells are composed of organelles that are made of proteins and amino acids. And all of these microscopic structures are going about their job, carrying out essential functions to allow the larger organism to function as well. Microscopes have shown us that none of us is an individual, that we are in fact more prokaryotic cells than human cells. Prokaryotes are single-celled organisms. And we require all of those prokaryotic cells to do whatever it is that they're doing in order to allow us to think of ourselves as an individual and to do what we need to do. So our world has been transformed and made so much more interesting through the microscope, through technology. In the best instances, it opens our eyes and it allows us to examine our biases and our assumptions. So this has very much been the case for myself and Kevin in terms of our research. We've been able to look closer, we can look underneath, we can look within, as we carry out our science in new ways, in ways that our advisors weren't able to work. So that's what we're going to talk about today, a bit about our research that we've been conducting, and we're going to highlight the new technologies that we're both using and discuss what that has brought to our research. So two of the most fundamental traits of modern humans is one, our unique ability among primates to walk on just two limbs, and our amazingly dexterous upper limbs and hands. There are so many questions about the timing and the reasons for our transition from arboreal and quadrupedal behaviors to bipedality, and from an upper limb that was used primarily for grasping branches overhead to one that is oriented towards precision manipulative behaviors. So before we dive into this, I want to spend a little bit of time orienting everybody to our human family tree, some of our hominin ancestors. So this is one presentation of our human family tree as it currently stands. And time is on the y-axis over here. And each of these bars represents a different species showing the approximate time period for which they live. So one thing that you can see really quickly is that there used to be a tremendous amount of species diversity, and within that species diversity, just a tremendous amount of organismal diversity. So as biologists, we know that variation is required for natural selection. And natural selection was certainly acting to influence the evolutionary patterns that we see here. So it was one of the mechanisms of evolution that resulted in our current morphology and behavioral profile. So the second thing I want to draw your attention to is this little blue box down at the bottom where it says last common ancestor. This represents the time period when the human lineage split from the chimpanzee lineage. So this is when we last had an ancestor in common with chimpanzees. And since this time, they have been diverging and evolving in their own unique chimpanzee direction. And we have been diverging and evolving in our own unique human direction. So Kevin and I are going to use the term hominin to refer to any species that's more closely related to us than they are to chimpanzees. Okay, so at the very top in red, you can see us. And we emerged about 300,000 years ago. So then going to the bottom, we have this group in dark gray, and these may have been among the first hominins. We don't know for sure whether they were hominins or where these taxa, these species fit into our evolutionary history, but some evidence suggests that they may have been hominins. So part of the reason that we're not quite sure where to place them is because the chimpanzee fossil record is shockingly sparse. There are only 2 fossils of chimpanzee ancestors. There are 2 incisors that were published in 2005. So we just don't know a lot about what the ancestors of chimps would have looked like. Anyway, one of the species from this ambiguous group right here is Ardipithecus ramidus. And you might have heard of it because it received a lot of attention in 2009 when it was published. And this individual received the nickname Artie. So Artie had opposable thumbs like us, but she also had an opposable big toe, not like us. And she is thought to have walked bipedally in kind of a wonky fashion. She also had really short, stubby thumbs, which is decidedly non-human-like. So then once we move up a little bit closer in time, we can see that there's kind of an explosion of species. There are lots of archaic hominin species, which are shown here in green. And most of them are attributed to the genus Australopithecus. They lived in different areas at different times between about 4 million years ago and 2 million years ago. And this is the group that includes the famous Lucy fossil. Lucy comes from a species called Australopithecus afarensis. And afarensis were small-bodied apes that were bipedal. We know that they were bipedal by the footprints that they left behind. You can see those footprints in the image on the right that are attributed to afarensis. So continuing up the time scale, we see next Paranthropus, which are megadont or hypermegadont species. This just means that they had massive teeth and jaws. And you can see that in the image at the bottom. The skull on the right is a human skull, and you can see its dental arcade. And then compare that to the Boisei maxilla on the left. So we take their massive dentition as a sign that they were eating like really crappy food, like bark and grasses. And then if we move to the left, the light blue group, these are so-called transitional hominins. These are basically morphologically intermediate between archaic hominins and hominins that are solidly within our own genus, that were solidly Homo. So this is KNMER 1813, who was a Homo habilis, a particularly nicely preserved example. And if you look at the skull, you can see it looks pretty human-like overall. But it has a really small cranial capacity. And if you flip it over, you can see it also has really large teeth. So in some ways like us, in other ways very much not like us. And then after those transitional hominins, we have pre-modern humans, pre-modern Homo, which is this last group in dark blue. And these species are very human-like in a lot of respects, but then some of them, or all of them, are different from us in some really important ways too. So the group on the left that I've highlighted here includes Homo erectus, and these, also Neanderthals. Members of these species are really, really, really human-like. Whereas on the other side of Homo erectus, we get some members of the genus Homo who are just kind of different than what we would expect. This includes Homo floresiensis, which was the Hobbit. Do you all remember when the Hobbit was published in 2004? That was like the most exciting thing that has happened in the course of my life. I wasn't associated with this at all. But the Hobbit was 3.5 feet tall, lived on an island. So things on islands either get really big or really small. The hominid floresiensis got small. They made stone tools, which I thought was cool. And then we also have Homo naledi here. Homo naledi has received a lot of attention recently. These may have been intentionally buried, not sure. But what you can see is that for most of the human lineage, our genus has been really speciose, or there has been a lot of species within our genus. And all of these species were presumably adaptively dynamic and figuring out how to survive in their own unique ways, according to their own anatomy and physiology and neurology. So it's really only within the last 15,000 years or so that we see a single species within our genus, a single species of hominins. Why that is something really fascinating to think about at another time. So now that we're all oriented and we're familiar with some of the celebrities of our human family tree, I want to focus in on the period that Kevin and I work in, which is about 3.5 million to 1.5 million years ago. So this is a particularly speciose time, as you can see on this image here. This is when there were a lot of hominins, especially throughout Central and Southern Africa, and many of them were living in the same region, if not in the exact same localities. So I'm going to get us started focusing on my research, which focuses on the upper limb and thinking about how did we develop a forelimb? How did we evolve a forelimb and a hand that's adept for precision behaviors, having come from one that was geared towards grasping trees and branches overhead. So fossil evidence demonstrates that we see a shift from curved fingers that may reflect greater emphasis on arboreal behaviors and a really short gracile thumb, this is Artie again, towards straighter fingers, as we see in Australopithecus afarensis or Lucy, towards an elongated and eventually a more robust thumb. How did we get from one to the next? And I don't mean to imply with these images or what I'm saying that this was a linear trajectory, nor do these species represent direct ancestor-descent relationships. These are just the trends that we see in the fossils as the fossil record presently exists. And these are all snapshots of what came before our own large, robust thumb, straight-fingered anatomy. So the prevailing thought is that our hands and really our entire forelimb is some combination of adaptation and exaptation to the production and the use of tools. And when I say exaptation, I mean a feature that developed for one purpose and has been co-opted and now used for another purpose. So for a variety of reasons, not the least of which being that they can enter the paleontological record, the tool use that has kind of become the most important or received the most attention in terms of what was selecting for our hand and wrist is stone tool production and use. So the thinking connecting the anatomy of our forelimb to stone tool goes like this. We know that quadrupedal behaviors place a lot of force and a lot of wear and tear on the forelimbs. All forms of quadrupedal locomotion are associated with large ground reaction forces, which I've represented by the blue arrow in this image. So this is the force that's exerted by the ground it also occurs in bipedal locomotion. So like think about when you hit your heel on the ground as you're walking or running, that's the ground reaction force. And over time, those forces are going to lead to injury, or they would have, except that we developed special anatomical configurations to help us resist that damage. So the same is true for quadrupedal animals. They have evolved anatomy to protect them from those high ground reaction forces. They've evolved this anatomy in their forelimb. Part of that anatomy is these really large, strong forelimb muscles that can be used to contract and counter those forces, those ground reaction forces, and to maintain joint stability and to help avoid injury. So here we're looking at muscle recruitment data from a baboon. And if you're used to looking at these kind of data, you would say to yourself, oh, that individual is recruiting their forelimb muscles really, really strongly to resist those ground reaction forces. So the ability to locomote efficiently and safely is paramount to the survival of an organism. This is so much so the case that the anatomical requirements for safe and efficient locomotion superseded those of nearly all other behaviors in regard to the anatomy of the upper limb. So that, combined with those strong ground reaction forces acting on the hand and wrist, meant that the forelimbs of our pre-biped ancestors were dictated almost entirely by locomotor demands. It took what I think of as a behavioral revolution to get the forelimbs of our ancestors to begin changing in a new direction. So our ancestors had to come down from the trees and begin walking across the landscape on two legs in order to free our hands and our arms and our wrists from their locomotor restraints. And that opened the door for a new behavior. And like I said, the behaviors that have received the most attention are those related to stone tools, to making and using stone tools. And the hypothesis is that our forelimb was changing in ways that allowed our ancestors to make and use stone tools in an energetically efficient and biomechanically safe manner. And perhaps because of the relatively large impact forces that occur at strike, tool production, the making of these stone tools, kind of edged its way to the fore as being the primary selective pressure that selected for our upper limb anatomy. So the strike forces, the strike impact forces, are thought to have taken the place of ground reaction forces in terms of their influence on the upper limb anatomy. And the changes that we see in our upper limb anatomy are thought to reflect adaptations towards placing the hand in space, also helping to resist those impact forces and helping to generate the muscular forces that are needed to hold onto your tool. And I want to take a second to familiarize you with these really impressive tools that I'm talking about, because most of the time, I think when I say stone tools, people think of arrowheads or of a recognizable tool, but that's not what I'm talking about 3 million years ago. I'm talking about little flakes of stone that you would pass over if you saw them, or maybe the rocks that they were chipped off of. They do not look very impressive. My brothers tell me this all the time when I'm justifying what I do. But their significance on our evolution cannot be overstated. You have to think of us as small-bodied apes, virtually defenseless, competing against lions. So when we had tools, we suddenly had weapons. We could also butcher animals quickly, removing that meat and taking it away to a safe place. So stone tools were really this watershed event in human evolution. I know they don't look like much, but they were a big deal. So through the use of motion capture technology and infrared cameras and some very, very gracious volunteers, We know a lot about how the upper limb is moving during the production of stone tools. My collaborators and I used these super fast infrared cameras to record the movement of reflective markers that we super glued to the skin of our volunteers. I told you they were very gracious. And the system that we used spits out the 3D coordinates of each of these reflective markers. So we recorded at 200 hertz, which means that for each of our 27 markers, the system records 200 sets of the 3D coordinates per second. That's a lot of data. We were able to very nicely quantify how people are moving. And so this is what the data look like. You can see the markers on the left, and they're spit to a skeletal model on the right. And the reason that it looks like it's like flapping its hand around in space is because we couldn't put reflective markers on the hands. The space is too small, so it just looks like a glowing glove. So we put those 3D coordinates into our scripts, and we were able to quantify a large range of biomechanical variables, such as angular change at various joints, and angular velocity, and torque, which is rotational force, and from that, work and power. And we found that while making stone tools, people move their upper limb the same way, that a whip moves. So motion starts up at the shoulder and also in the trunk, in the large, powerful muscles that are able to generate a good deal of power. Motion then moves down from one limb segment and joint to the next. And as it does so, it's picking up velocity. And this results in significantly greater linear and angular velocities at the wrist than the limb could reach using any other motion sequence that we know of. And you can see these high velocities getting higher as we move from the shoulder to the elbow to the wrist here. And this motion strategy relies heavily on interactive torques, influencing one joint after the next in order to move each successive limb segment. So that means it's achieving these high velocities in an energetically efficient manner. And you can see that here. So we're looking at work produced at each joint. That's shown in red. I'm sorry if you are colorblind. I didn't think of that until moments ago. But if you look at work, it's not what you would expect given the velocities that we're reaching. So look at the elbow here. The elbow is moving pretty fast and it's producing a good amount of work. And it's passing that work on to the wrist, which is moving the fastest and producing virtually no work. So the wrist can move as quickly as it does because it's moving like the end of a whip. It's benefiting from all of that rapid motion that's occurring at the elbow. So this was great. This was well and good. It was really interesting to see how our limb is moving and to realize that this benefit exists given our anatomy. And it was also cool to realize that the fossil evidence that we have suggests that our hominin ancestors could have moved in the same way. So it could be that we landed on this energetically efficient way to make stone tools. So the highlighted region here shows us approximately when we begin seeing the types of tools that I'm talking about in the archaeological record. This is it was just pushed back to 3 million years, 2.5 million to 3 million years ago is when we start seeing these early tools. And this is also the time period when we see a significant jump in brain size. So given the nature of the fossil record, one of our best indicators, if not our only indicator of cognitive capacity, cognitive ability, is cranial capacity or the size of the skull. So part of the thinking is that the biomechanics of stone tool making and using, we're not only pushing our upper limb anatomy in a new direction towards greater precision, but the thinking goes, this behavior may have also been selecting for greater cognitive capacities. Or on the flip side, it may have been enabled that our ancestors may have been able to produce and use stone tools because something else was selecting for greater cognitive capacity. Either way, the ability to make and use stone tools is frequently regarded as requiring and indicative of fairly complex motor strategies and cognitive complexity. But lately I began to think about this a little bit more and ask myself if this is actually the case. Is this motion pattern really that complicated compared with what we see in other primates? So in the early and mid 1900s, a researcher named Bernstein argued that complex motions are those that make use of all 6 degrees of freedom that are available to them. So degrees of freedom is a term for independent motion available to an object around an axis of rotation. Some joints, like the shoulder, can move through all 6 degrees of freedom. Other joints, like your elbow and your wrist, may be limited to 2 or 3 degrees of freedom. So one way to compare the complexity of 2 different motion sequences is to consider the relative degrees of freedom at each joint, the number of joints, and the number of joints that are being coordinated in order to carry out that motion. So I've been thinking about my assumptions and biases, and I started thinking about those in regard to the biomechanics of underlying this early tool production. And I started to wonder whether that upper limb sequence is really that complex compared with other motion sequences that we see in other primate species. And is it really indicative of cognitive complexity as well? So when we think about tool use in other species, or in other primate species, we probably think about nut cracking in chimpanzees. They are our closest living relatives, and they're widely known to use hard objects to crack open nuts. So they seemed like the obvious comparative choice, but it's the tool use of these little guys, Sapajou libidinosus or capuchin monkeys, that really caught my attention because of the mechanics of it. So nut cracking in capuchins is a full body motion. They hoist a rock 60% of their body length. And in doing so, they're coordinating, they use both hands, so they're coordinating 3 joints in their upper limb and 4 joints in their lower limb on both sides of their body, moving through somewhere between 6 to 2 degrees of freedom each. And they're often using their tail for balance as well, so they're having to coordinate all of those joints. Again, we know all of this. How these little capuchins are moving has actually been quantified because of advances in motion capture technology. So before, most of this motion sequence was known simply qualitatively, and now we can quantify it. The research that I'm about to highlight is not my own, but I just think it's so cool. So compare what capuchins are doing to the tool production swing. Ethnographic evidence suggests that this was a seated behavior. It doesn't take that much force to knock a flake off of a stone. It's a light duty activity, so there's no reason to involve the lower limb. So if we agree that tool production is a seated behavior, we're looking at movement of only 3 unilateral joints. The shoulder, which modern tool makers move through 6 degrees of freedom, and then the elbow and the wrist, which both probably move between 2 and 3 degrees of freedom. So coordination of 6 degrees of freedom at the shoulder is nothing to turn your nose up at. Your shoulder is a really complicated series of joints. But some of our more recent work suggests that there are actually advantages to eliminating movement at the shoulder while you're making stone tools, and instead driving from the elbow. So this is another figure similar to what I showed earlier, showing angular velocities and work. And you can see that whip-like motion sequence is in effect as we move from the shoulder to the elbow to the wrist. And when we looked closer at our data, we realized that our subjects were conforming to one of 2 biomechanical strategies. And I should say that all of these subjects, bless their hearts, are like expert tool makers. So they're people who spend a lot of time making stone tools. And most of the time they're not making these flakes that I asked them to make. They're making really, really beautiful like works of art. And I found them on the internet and flew them to Washington, D.C. and the rest is history. So when we looked closer at these data, we found that people were conforming to one of two strategies, either one dominated by the shoulder or one dominated by the elbow. So in this elbow-dominated strategy, people are actually kind of locking their arm by their side and generating tremendous angular velocities, at the elbow, moving from flexion to extension. And when they do so, they're generating just an outrageous amount of work at the elbow, which is then being passed on to the wrist. And those beneficial torques are making the wrist in the elbow-dominated strategy reach really high linear and angular velocities. And that's resulting in higher strike force. So our preliminary analysis shows that when tool makers are using this elbow-dominated strategy, they're more than twice as efficient as those that are relying on the shoulder-dominated strategy in terms of the number of strikes per flake detachment. So this is likely due to two things: the increased strike force, and also the fact that if you eliminate motion at the shoulder, you're probably a lot more accurate. Small movements at your shoulder are going to result in large movements downstream. So if you can just lock that down and eliminate this whole complexity, you're a lot more accurate. So what we took away from this is that this biomechanically simpler strategy seems to have some significant advantages in terms of energetic and temporal efficiency. Another variable to consider is that capuchins use rocks that weigh up to 50% of their body mass when they're cracking open nuts. Archaeological evidence suggests that hammerstones, on the other hand, weigh on average 1.5 kilograms. Female capuchins are often carrying their juvenile on their back as they're cracking open nuts. And some capuchins get so into it that they jump up off of the ground in the course of this nut cracking swing. And a large portion of swings involve lifting the rock above your head. So think about what's going on here. They are bending and then lifting, sometimes with an infant on their back, a rock that weighs 50% of their body mass, 50% of your body mass above your head. So there is nothing simple about what these capuchins are doing. Tool use is really widespread in the primate order. And so this strongly suggests that the foundations of tool behaviors long predate hominins or even hominids, any of the apes. Percussive tool use in particular is also present in a lot of species, also suggesting that this type of behavior was present long before humans were on the scene. So when looked at in this context, it's hard to argue that this possibly seated, certainly unilateral upper limb behavior really warrants the designation of motor or cognitive complexity, especially when we compare it to the full body nut cracking swing of capuchins, during which stones at times weighing 50% of their body mass are hoisted an average of 60% of their body length. So this all nicely illustrates how advances in technology and motion capture have pushed my own research forward and enabled me to examine my own biases and my discipline's biases and assumptions. And we now understand that modern humans and likely our hominin ancestors possessed anatomy that allows us to engage in this really important behavior in a biomechanically efficient and safe manner. But maybe tool production doesn't belong on the complexity pedestal that we've placed it on. Maybe it's really no more complex biomechanically, cognitively, than behaviors that we see across the primate order. So now I'm going to turn things over to Kevin.
All right, quick check. Is my microphone working okay? All right, we're good. Perfect. All right, so now I'm going to shift our focus from hands down to feet in order to think about human locomotion. And first of all, why is locomotion so interesting or important? Well, locomotion is integral to the ways that any animal exists within and engages with their environment. It's one of the most fundamental reflections of natural selection. If we look at our closest living relatives, the apes, each species has a unique way of moving around its own environment. Orangutans and gibbons climb in trees in two very different ways. Gorillas climb trees to varying extents, but they mostly knuckle walk on the ground. Bonobos and chimpanzees have their own ways of knuckle walking on the ground and their own special ways of climbing in trees too. Among these apes, us humans are very different because we walk and run upright on two limbs instead of four. And our peculiar manner of bipedal locomotion is considered a defining feature of modern humans, and how we came to walk this way is one of the big questions around which our discipline is built. It's one of these questions that's been around since Darwin's time. Now, I want to step back just for a second and refer to this image that Erin Marie kindly introduced and point out the time period that I study most often. I tend to focus my research on this particular time window from about 2 to 1.5 million years ago. And this interval is really interesting because this is when several of those archaic hominins are sort of fizzling out, and pre-modern Homo is just emerging. The species that's just emerging here in this time window is Homo erectus, and Homo erectus is probably as strong of a candidate as there is right now for being a possible direct ancestor of ours. Now, my own field research is concentrated in Eastern Africa, and so I'm normally thinking the most about 4 of the 6 species that fall into that time window, because the others are known only from Southern Africa. And this is a very interesting time and place in our evolutionary history because these 4 hominin species here might have overlapped and sometimes even lived on the same landscapes as each other, and they seem to come from 3 very different adaptive grades. For us today, again, it probably seems totally bizarre to think about living right next to members of a different species, much less multiple different species that are very closely related to us and probably similar to us in a lot of ways, but also adapted to very different ecological niches. So in this time window, we know of Homo habilis and Homo rudolfensis over here, who share relatively small bodies and brains. They have kind of intermediate teeth. They're not as large as Paranthropus teeth, but not as small as ours either. And from the neck down, they look a lot like those archaic hominins that we know of from the time period before this one. Then there's Paranthropus boisei in here. They're characterized by, again, small bodies and brains in relative terms. They've got these enormous teeth and jaws that suggest, again, that they're probably eating very different foods than we do and than the other hominins are around this time. And again, from the neck down, they still look very similar to those species those archaic hominin species that predate them. And then finally, Homo erectus emerges during this time period, and all of a sudden they look very human-like. They look human-like in their body size, their body shape, they've got large brains, and their teeth look a whole lot more like ours than any other hominin that was around up until this point in time. This is also thought to be the first species to walk and run in a modern human-like way. So this is a very interesting time period for people like me who study the evolution of human locomotion. Now, I also want to spend just a second thinking about how people typically study locomotion in the fossil record. This is a famous and remarkably complete Homo erectus skeleton that's about 1.5 million years old that was discovered near Nariakotomi, Kenya in 1984. And it looks human-like in its overall morphology. And based on that human-like skeletal morphology and what we know about the ways in which skeletal form relates to function, people have hypothesized that this individual, and probably the species on the whole, would have walked and ran in very similar ways to modern humans. Digging a little deeper into how people have arrived at this kind of hypothesis, researchers like myself who are interested in what we call functional morphology search for the various ways in which functional signals can be inferred from usually skeletal anatomy. Before anything can be inferred from fossils, a huge amount of experimental research is necessary to demonstrate how functional signals are reflected and or recorded in the anatomies of our bones. And so the examples here are just some of many different experimental studies that have shown how things like joint morphology, trabecular bone anatomy, which is like your inner bone anatomy, or the cross-sectional geometry of cortical bone, the external shape of your bones, might record the capabilities of and or the mechanical stresses experienced by these structures during their corresponding organisms' lives. Now, these kinds of studies reveal those mechanical connections between form and function, which then provide the informed basis by which paleoanthropologists can look at fossils and begin to understand how our ancestors and relatives moved around their worlds. In my own research, I've also tried to think about what other kinds of evidence might exist, which we can also use to understand the evolution of human locomotion. And one of the rapidly emerging lines that I've spent a lot of time focusing on is fossil footprints, which are essentially these snapshots of locomotion itself. Within each of these snapshots are direct evidence of both foot anatomy and the way these individuals were walking at specific moments in time. And those pieces of information can be really valuable for understanding locomotion throughout human evolution. There haven't been too many opportunities historically for intensive research into this kind of evidence, though, because for decades, these hominin footprints were extremely rare. The two sites on the screen here were both discovered in the late 1970s, I believe in the same year, actually, in 1978. There's the famous site of Laetoli in Tanzania on the left, which preserves a set of 3.66 million-year-old footprints thought to belong to Australopithecus afarensis, Lucy species. On the right, there's another site at Kubifora, Kenya, that's about 1.4 million years old, and it preserves a not nearly as extensive set of footprints from a single hominin that was walking here, and we don't know for sure which species made those ones. So for about 3 decades that followed these two discoveries in 1978, these were the only sets of early hominin footprints that were known that predated modern humans. There weren't a whole lot of data to work with. But that picture has really changed in the past 15 years or so. As I was entering graduate school in 2009, this discovery was announced in Science of these 1.5 million-year-old hominin footprints that were discovered near Illoret, Kenya. My PhD advisor was a part of that project, along with working with a large team of US, European, and Kenyan scientists. I was really interested in studying locomotion and the evolution of these early members of our own genus. And they believed these footprints might have been made by Homo erectus. So I was really excited to be able to start contributing to this project as an early PhD student. Now, when I first went to this area in 2010, we were continuing to excavate that site that was published in 2009. That's designated by the blue star on this map. Here we're looking at an aerial image with a grid that's overlaying the fossil deposits in that Illoret area. And if you look at the scale on the left, you see that this is roughly 1.5 square kilometers or so that we're looking at. So in the few years that followed, we also started looking in this area for more of those fossil footprint surfaces. I was really fortunate to be able to work with Kay Berensmeyer, the scientist who had discovered those Kubifora footprints back in the 1970s, and she trained me and others who I was working with on how to identify the locations where these kinds of fossils are likely to be preserved. So we did a systematic survey. That's why there's a numbered grid sitting on top of this map in which we would just randomly choose a number, go to that location, see if we could find a fossil footprint site there. And so through these surveys, we ended up finding that these kinds of sites were just all over the place here. So by 2014, we had found 25 different sites that together preserved more than 475 fossil footprints. Among these were 7 sites that preserved a total of over 100 fossil hominin footprints. Now, those numbers might not seem very large to those of you who don't work in this field, but for those of us who work with fossils, this is like an order of magnitude larger than what we're typically dealing with in one of our samples. So we estimate that those 101 footprints made across these 7 distinct sites were made by more than 15 hominin individuals who inhabited this area around 1.5 million years ago. And we hypothesize that most of those footprints were made by Homo erectus. We say this because of their size. I mentioned earlier that Homo erectus tended to have larger body sizes compared with the other hominins that were around during this time. Some of these footprints are longer than a men's size 13 shoe. So for at least those very large ones, we estimate that Homo erectus most likely made those. Now, even more recently, our ongoing field research in the Turkana Basin, which is in northern Kenya, has continued to be extremely productive. We're still finding lots of new and very exciting things. This photo is from 2022 when I was working with colleagues here to excavate another fossil footprint site at Kubifora that's slightly older than any of the others we had found previously. And here you can see that after excavating, we uncovered another amazing set of these fossil hominin footprints. There's one very long 13-step hominin trackway going from left to right across the screen here. There are isolated footprints from 3 more individuals who were walking in a roughly perpendicular direction. And there are footprints of other fun animals here too, like these massive storks that were potentially like 6 feet tall or more. They were walking all over this site too. There were bovids, equids, all sorts of other animals here too. And we're still working to publish a paper on this site. It's currently in the peer review process, so major buzzkill, I won't be able to share more about this one today, but maybe at a Café Sci in the future. Now, I've mentioned only a couple of sites where I've worked. I want to emphasize that a whole lot more has been found in recent years too. Within just the past 5 years, we've seen this really amazing surge of new discoveries or new analyses of fossil hominin footprints from all of these locations around the globe. And they span an impressive range of space and also time, going from about 3.66 million years ago all the way forward to about 10,000 years ago. And these sites are giving us this influx of new data that are now available for understanding hominin locomotion at many different intervals over the past several million years. So finding these new fossils is always exciting. We know that they're giving us these amazing snapshots of our extinct relatives, but then we also have to ask the question of how can we then use these data to understand the evolution of human locomotion? And to do this, we have to approach this as a question of functional morphology. We need to understand how the morphology of the footprints reflects or records the functional pattern, the locomotor pattern in this case, that produced them. So here's where technology comes into play, because new technologies that were not available until very recently, certainly not available in the 1970s, have allowed us to lift up the hood and study exactly how a footprint is formed. So these 2 researchers who are now close colleagues of mine, Peter Falkingham at Liverpool John Moores University in the UK and Steve Gatesy at Brown, had started just over a decade ago using biplanar X-ray and computer simulation to observe and model the process of footprint formation in birds. They hoped to better understand the fossil footprints of theropods, which are bird-like dinosaurs. As they were developing these techniques and working on this paper, I reached out to them and we agreed it would be really exciting to try to do this in humans. And so over the course of 9 years now, that's what we've been doing. We've been developing and implementing biplanar X-ray methods that we can use to study the mechanics of how human footprints are made. And there were many prior iterations of this. Again, it's been 9 years that we've been working at this. It took a whole lot of trial and error in that time. But in our most recent experiments, we capture a high-resolution 3D scan of someone's foot. And if you can see them in the audience, there are little black dots drawn here. Those are the locations where we place tiny little radiopaque markers that we stick on people's feet with medical adhesive. We're not using super glue in our experiments. Subjects then walk along a foam-covered trackway with a radiolucent substrate that we place in the middle within the volume of space that can be observed with this biplanar X-ray equipment. So biplanar meaning you're capturing X-rays from 2 different sort of overlapping fields. People make a footprint in that space where we can capture biplanar X-ray video. We also then take a high-resolution 3D scan of the footprint that they make. Using custom software, we can then accurately track the 3D motions of the foot while it made that specific footprint. And then with these data, we can build 3D animations of the entire scene of these trials, which includes the foot moving and deforming realistically in sync with those observed marker motions. We also accurately register the 3D model of the footprint that was made here, so then we can observe and analyze how the foot was moving and deforming relative to that footprint it created. Then we've also used new technology to quantitatively analyze the deformation of the substrate itself. And here the credit goes to my colleague Peter, who designed this approach where he's using supercomputers to model the physical behavior of every individual particle within this volume of substrate, including all the interactions with the foot and with the particles around it. We could use our real-life experiments as a source for validation to then accurately simulate the substrates that we used in the lab. We could drive our animated foot through this virtual mud using the motion and deformation data that were actually observed in our experimental trial. And we eventually landed on a method that produces simulated tracks that are exceptionally close to the true experimental ones. In this case, the average difference between the two was less than 2 millimeters. Most importantly, though, this simulation approach, it allows us to study exactly how substrates and feet mechanically interact to produce these footprints. And that is what has become a game changer for us because it gives us this path to understand how the anatomical and biomechanical signals are actually getting recorded in the morphology of footprints like the ones we know of from the human fossil record. So with the rest of our time, I want to just highlight some of our latest results, which have come from our development of this new technology. And zooming out for a second, if we think about key questions and hypotheses about the evolution of human locomotion, biological anthropologists and lots of other scientists have focused a lot of attention on our arched feet. Longitudinal arches are often thought of as one of the most important evolutionary innovations of the modern human foot. No other ape has a foot that's arched like ours, and classic papers describe this structure as essential for allowing our foot to act as a stiff propulsive lever during bipedal locomotion. Now that view has changed over time and become a little more nuanced. Other people have suggested that longitudinal arches might be more important for the way that our foot stores elastic energy during endurance running, but one way or the other, our arched feet are thought to be integral to the way that we walk and run. Now, it's very hard, though, to use fossil foot bones to understand much about the evolution of arches, not only because samples of fossil foot bones are typically very small and fragmentary, but also the arched configuration of our feet has a lot to do with the anatomies of ligaments and other kinds of soft tissues that do not typically fossilize. Instead, evidence that's long been thought of as most reliable for evaluating the arches of feet comes in the form of fossil footprints. Footprints like this 3.66 million year old one from Laetoli get cited routinely as clear, indisputable evidence of arched feet going back millions of years. And that conclusion is based on some intuitive rationale. These footprints have a raised area that's about midway along their length. It looks a lot like what you would see if you lifted up your own foot and looked at the bottom of it. But if we're being honest, we've never really known what this arched shape of footprints really means. It could resemble the anatomy of the foot, but it might not. Footprints are formed through this very complex interaction between a foot that's deforming and moving through a deforming substrate. So it's very possible that something more complicated is happening. So we decided to investigate this question for the first time using the approaches that we developed for studying the mechanics of how a footprint gets made. We used those biplanar X-ray and 3D animation techniques that I just explained to directly visualize and analyze how feet and substrates interact while a longitudinally arched footprint is being made. What we found is that the arching of footprints, it actually has a whole lot more to do with overall lower limb kinematics, the way your entire lower limb is moving, than it does foot anatomy. So when we walk bipedally, we strike the ground with our heel, we smoothly rotate our foot so that it's flat, and then we push off with our forefoot. This motion pattern increases the effective length of our lower limb. We walk with what's called an inverted pendulum approach. And by rotating our foot in this way, we see that the pivot point of this inverted pendulum here is actually located below the surface of the ground. And so this is part of what helps us to walk in an energetically efficient manner. In our experiments, we could visualize the foot's heel-to-toe rollover by mapping the trajectory of a pivot point shown in black here that separates the parts of the foot moving up, indicated by the red vectors, from the parts moving down, indicated by the blue vectors. And that pivot point, it moves gradually from heel towards toes while your foot is in contact with the ground during a step. Now, as the foot moves in this fashion within a substrate that can deform, that substrate's going to move down as the various parts of the foot move down. And that pivoting motion eventually generates a longitudinally arched footprint. As a theoretical test, we also applied this advancing pivot rotational motion to just a rigid and flat block, essentially like a virtual brick. And through our substrate simulations, we could see that even that brick could produce a longitudinally arched footprint as long as it used that particular motion pattern. In other words, it's clearly the motion pattern, not anything about foot anatomy, that's responsible for generating longitudinally arched footprints. After making that discovery in the lab, we then came up with ways to quantitatively measure the arches of footprints using 3D animation tools. And we published and shared those software tools earlier this year. What these methods do is they provide us with a way to actually obtain a comparative measure of foot kinematics, of how the foot was moving through the mud as an individual was walking and making a footprint. So the pattern of foot kinematics that humans use, the pattern of foot motion that we use, generates a strong and consistent signal that gets reflected in the arches of our footprints. Here I'm plotting a logarithmic fit based on human footprints I've collected in experiments over the past decade or so. On the y-axis, we're looking at something called relative arch volume, RAV, which is essentially a normalized measurement of arch height. On the x-axis is relative depth, and we see that human footprints, they tend to get more arched as they get deeper. This is because that heel to toe rollover, it gets exaggerated when you're walking on something that can deform beneath you. But eventually that exaggeration sort of levels off. You can only go so far. Now this human sample, it includes footprints from habitually shod people. There are footprints that I collected in the lab here in the States. Also footprints that I collected in the field from people who are habitually barefoot. So there's a really wide variety of foot morphology that's represented in this plot. But despite that, humans tend to follow very similar patterns. I can even throw into the mix some 400 to 500-year-old human footprints from a site at Walvis Bay, Namibia. And we see that even these fit very well within the same kinds of trends that we've been observing in the lab. So we can be reasonably confident that what we're capturing here is really a signal of how the human foot moves. Now, in a paper last year, we then showed how those 3.66 million-year-old Laetoli footprints, including footprints from two different trackways, one called G1, the other called S1, those are shown in green here. They deviate from that human trend. They're generally flatter than what we'd expect for similarly deep human footprints. And we explained in that paper how this is a strong indication of differences in their foot kinematics. We can say with a reasonable bit of confidence that they were walking in a different way than we walk today. We further highlighted how this other footprint from a different area, but the same geological level at Laetoli, so the same age, this other footprint is shown in pink on the plot here. It's different and much flatter than the other ones from Laetoli. And this was exciting because it supported another paper that I worked on with colleagues where we found that two different patterns of hominin bipedalism are evidenced at Laetoli. That's really exciting because it opens up all sorts of new ideas and questions about whether and how these hominin species who must have lived right alongside one another might have interacted, competed, and potentially influenced each other's evolution. Now, if I had a sample of those 1.5 million-year-old footprints that my colleagues and I excavated from sites near Illoret, Kenya, we see that these follow very closely the arching patterns that we see in modern human footprints. This tells us that these hominins had feet that probably looked a lot like ours and also moved a lot like ours. That's a picture actually of my own foot next to one of these footprints, and they look reasonably similar to one another. So this gives us a rare piece then of direct evidence that supports this hypothesis that significant changes to locomotion occurred in Homo erectus and differentiated that species from both their contemporaries and their predecessors. These changes to locomotion might have been associated with a whole series of adaptations that characterized later members of the genus Homo, including our own species, Homo sapiens. So to wrap up and summarize quickly what we've found, first, there are a whole lot more fossil footprints out there than previously thought, which is exciting because that means we have a whole lot more data to work with all of a sudden. Then with these new technologies and new methods, we've been able to learn new things about the evolution of human locomotion. Our analyses are telling us new things about the ways in which extinct hominins moved. But then we're also learning new things we didn't expect. For example, those analyses that have allowed us to identify locomotor diversity within some of these sites. We couldn't do that previously, but that has huge implications for testing different kinds of hypotheses, again, about the nature of interactions and potentially competition in the evolution of our fossil relatives. And there's still more that we're learning every day too. So with that, I want to thank a long list of collaborators and co-authors here who were involved with different aspects of the research I presented today. That list includes several, but not all of the many, many students who have worked with me on all aspects of this work. So I want to thank all of them as well. Of course, I want to thank the organizations that funded this research. And Erin Marie has a list of collaborators and funders that she would like to thank as well. And thank you again for inviting us here. And thank you to all of you for coming and listening.
All right, thank you so much, Erin, Marie, and Kevin. We'll move into Q&A here shortly. Just a reminder that if you are here on site, you'd like to ask a question, Samuel will bring you that microphone, so please wait for that. Again, if you are on Zoom, you can use the Q&A button at the bottom of your Zoom window. And I already see hands going up, so this is going to be great. So why don't we bring both of you up on stage and we will get the questions coming.
Hello. First, let me say thank you to you both. It's been very engaging and interesting to me. My question is to Kevin. As a former marathoner, I know that modern humans run differently. If you watch runners and how their feet strike the ground, you will notice that some runners, such as myself, used to hit the heel first, and then the motion rolls along the outer edge of the foot to the toes. Other runners strike the ground differently. I've watched many of them, and go heel to toe without that pronounced side rotation. One's used running shoes may and sometimes indicate the pattern of the runner's footfall. Have you considered this difference in your research? And thank you.
Yeah, that's a great question, especially relevant given that we're just past marathon weekend now too. So yeah, running is interesting. You know, there's a lot of variation in the ways that modern humans engage in both walking and running. Running perhaps, you know, a little bit more pronounced maybe. Like you said, there are many different ways that people might strike the ground with their foot, different ways that they might transfer weight over their foot. And so they're accomplishing the same sort of end goal, but through slightly different biomechanical patterns. I think there's a lot of variation because people's bodies are different. People's, you know, your individual anatomy, if we were to survey the room here, we'd have a huge spectrum of, you know, different skeletal morphologies, different muscle morphologies. The ways people's muscles work would be different from person to person as well. The compliance and ligaments and things of that nature would also differ from person to person across the room. And so it is absolutely something that we have to consider and take into account as we're doing this kind of research. And so it's one of the reasons why we're very concerned with, like, when designing an experiment, like the biplanar X-ray experiments we're doing in the lab, we're trying to sample as many people with different kinds of foot anatomies as possible so that we can try to get some reflection in our modern human data sets of that kind of variation. The challenge when you go back into the fossil record and you have a relatively, you know, you're limited, you have what you found to work with, and the sample sizes are typically much smaller. So in many ways, you know, we have to remain cognizant of that sort of variation when we're analyzing that type of data. But it's much more difficult to do when you have, you know, you might have only a sample of 5 foot bones or 5 footprints that you have to work with from a given site. So it is for sure, you know, a somewhat limiting factor, or at least a challenge that we have to be aware of constantly. very good question.
All right, we've got some questions coming in online as well. These are sort of chronological questions that were asked during the talk. So I believe this first one is directed at Erin Marie. As you were talking about chimp fossils being so scarce in the fossil record, Kitty Mama wants to know if we have any reasons for why they're so sparse, those fossils.
Do we have any legitimate, we do have reasons, but do we have any like, deposition, did you say? You'd speak to this. I don't know why I was going to say because it's not as sexy to report on ancestral chimps as it is on ancestral humans. So every fossil that's found is, it is definitively, according to the researchers, hominins. So like, one of the really early ones, Chidensis, is maybe not hominin, but the researchers definitely want it to be a hominin. Although at this point, chimps are so sparse, that fossil record is so sparse that is probably just as sexy now. But now you give the depositional reason.
Yeah, so another reason as well, I mean, thinking about where chimpanzees and
Right.
other non-human great apes tend to live, so they tend to live in heavily forested environments. When their bones end up in the ground in those heavily forested environments, a lot of times the sort of soil conditions are not optimal for fossilization to occur. And so they're kind of like, yeah, you're in this situation where these animals almost certainly, you know, did exist. There are almost certainly fossils somewhere. But the number of fossils or the number of those apes that make their way into the fossil record is probably significantly smaller than the number of fossil humans that would make their way into the fossil record, just because of where they tended to live and the types of environments in which their bodies would have eventually been deposited. Those are also like thinking about areas that were forested way back in the past, like many of those areas are still very heavily forested. So it's also a really, really difficult place to go and look for those fossils too.
Yeah.
As opposed to the areas like I was showing in Kenya, these areas where there's enormous amounts of uplift that have exposed the fossil bearing sediments. And also those are very open areas that are sort of easy to search over.
Good answer.
Hi, yeah, thanks for talking. The technology you guys were talking about is really incredible and exciting to see in person. I think it seems like it would be expensive. Has the research become cost prohibitive, and has it changed how you seek collaborators as you're looking to do more research?
Yes. So Kevin's research is so much more expensive than mine. And mine was horrifyingly, I mean, the lab that I collected my data in was a shared lab. It was cobbled together through a number of grants and shared by across the university. So you can't, I mean, most of us don't have individual facilities of this nature. Collaboration is key. We always tell our students, collaboration is key. You cannot do it alone, right, guys?
Yeah, I think for mine as well, it's the same thing. You know, that's why much of that research I mentioned, it was taking place at Brown University. That's where they have a facility that was pieced together through funding from the Keck Foundation. It's a large shared lab that many different researchers are using, and they also sort of open that space up for other researchers to come in and essentially like pay rent, you know, pay lab fees, which are way, way, way smaller than what it would cost to build a lab like that of your own. But it's a great opportunity to be able to go and use that specialized equipment.
Yes, thank you. This is a question for Erin from your findings on the making tools from stone that mechanical action did not necessarily, if I'm understanding this correctly, require as much cognitive understanding. If that's the case, then what does that mean to the significance of discovering stone fossils as to what that meant in the, the evolution of
What,
us.
can you ask that last part again in a slightly different
Well,
way?
I'm
I'm not
just
sure I understood.
Curious, based on the results of what you have so far, does that change the way we look at fossils records as to what they mean?
Sure, right. Well, I would say that it doesn't necessarily change. It depends on who you talk with, I guess. Because our data sources are so sparse, we attribute a lot to each piece of information that we squeeze out of a fossil. So for example, I didn't talk about this aspect of my research, but another thing that I do is study muscle and ligament attachment sites on bones that leaves this morphological indication, maybe, that a muscle or ligament attached there. And so there are some muscles that are so important, especially those related to our hands, that evidence that those muscles existed in a given species is taken as evidence that the members of the species could engage in certain behaviors, specifically stone tool production. And if they were engaging in stone tool production, maybe they were living in complex social structures, and maybe they had language, and so do you see what I'm like? It's just this, we pack a lot in, whether or not we should, to small pieces of information that we squeeze out of fossils. So not everybody does this. Some people are far more cautious. So whether or not the idea that perhaps this motion sequence is not as biomechanically complex as other people think it is, that may or may not be impactful for how people are interpreting the relevant fossils. There's just a lot of disagreement.
So you guys briefly talked about how a lot of people like just wish that the fossils they find are a part of the Homo genus. I was wondering, like, are there key distinctions morphologically? They probably have to be more morphological since these are fossils, between something like Homo erectus and Homo sapiens, which lead you to believe that these are two different species. If that makes sense, I can try to reword it a little bit.
Mateo, I couldn't hear you at all.
Oh, sorry. Are there key distinctions between Homo erectus and Homo sapiens which make you believe there are different species?
Yeah, I mean, there's still noticeable difference. So we think of Homo erectus as like representing this kind of, very dramatically human-like general anatomy compared with everything else that was around at the same time, but it's still quite a bit different from us. Like you would notice for sure if Homo erectus walked into the room, like you would notice something was different about them. I mean, their like cranial capacity was still a good bit smaller than ours, even though it was quite a bit larger than everything else before it. Teeth are still different, larger than ours, but again, like smaller than everything before it. So it really looks like kind of I hate to think of things in a linear, directional fashion, but it's almost like an early step towards something that all of a sudden looks much more human-like. And so, yeah, not to imply any like linearity necessarily. Yeah, generally similar, but still like slightly different enough that you would notice.
All right, so we've got another online question here. This one comes in from Virginia, the person, not the state. And Virginia wants to know, how can the footprints that you study, which are so old, stay preserved for scientists to discover in modern times?
Yeah, that's a good question. So, I mean, footprints, so these footprints that we're studying, so they're made in mud, first of all. So when they were made 1.5 million years ago, they were made along a sort of lake or delta margin along the shoreline of water where the water table was sort of high enough, where this was not too soupy, it's like that perfect zone of mud that you have to walk through where you can make nice toe impressions and things like that without the water sort of filling in the footprint immediately and making it collapse. And it's also not too far inland where it's too dry to actually leave a noticeable mark. So you have to make the footprint in the right place, but then you also need a particular set of conditions to lead to it being deposited in the geological record, so it's then preserved for 1.5 million years. What that typically takes is pretty soon after those footprints are made, like within hours to a couple of days usually, you need to have some other sediment come in and be sort of washed over that to bury it. And so the places where this happens, like it was on a lake margin where the margin of that lake sort of transgressed and regressed, moved in and out continuously for hundreds of thousands of years, actually, in this particular place. That's why we found so many of these sites in that one particular area. It's an area where you had these sort of water margin environments that were being formed. They'd be there for a few hours, a couple of days. Then the water margin would move and bury those with some different kinds of sediments on top. That would happen over and over and over again. So you end up with this, you have very rapid sedimentation, I should say. So really rapid accumulation of lots and lots of sediments on top of one another. And so then you very quickly have this very deep sedimentary package that includes several footprint layers within it. That doesn't happen everywhere. I mean, it takes a certain set of geological and kind of hydrological conditions for that to occur. So this particular place seems to be kind of extraordinary in that regard. There are other conditions too, like those Laetoli footprints. Those were formed in volcanic ash. When volcanic ash falls and if it rains and that volcanic ash gets wet and then it dries out. So it gets wet, you make footprints in it, and then the ash dries out. It turns to something that is almost like concrete. It's a more fragile version of concrete. It's not like the concrete that we typically think of. But yeah, it's very resilient. Again, the time scale is something like hours to a few days or so that process has to occur without being disrupted for that to make its way into the geological record. So yeah, it takes a particular set of circumstances, but if you think of all of the footprints that you've ever made in your lifetime, like there are a good amount of these that get produced over time. And so, even if it's very rare for them to be preserved, we still have a decent sample of them.
Great, thanks. Okay, a question for Dr. Erin. You mentioned about the modern stone workers there, and you found them on the internet and brought them to DC. Where are they from and what are they actually doing now? I mean, that just fascinated me.
Yeah, it fascinates me too. So I mean, there is a huge community of people who make these ancient stone tools. They're called nappers, and they go to conferences called nap-ins. And I think that, when I was collecting these data, I was one of the first people to quantify this behavior. And so they were just so excited that someone was paying attention to their art form. And so I would, the wonders of the internet, I just went on napping forums. It helped that my undergraduate advisor, John Whitaker, literally wrote the book on napping. So he introduced me to people, and then I just kind of lurked around these chat rooms. That sounds so terrible. I flew out dozens of people to DC, some of them were archaeologists and professors. one gentleman who came out had never been on an airplane, he had never been on a bus, he had never been on a train. So he flew to DC. I picked him up on the metro and then we took the bus up to his hotel. I mean, they would take 2 or 3 days off from work to come do this. They were so and then we would super glue markers on them. They were so generous with their time. I just, what do they do normally? So there's really a tremendous amount of diversity in the community. But if you like Google Nappins, and you will find Kevin's colleague, Steve Gatesy, who he showed a picture of, now regularly attends these conferences. Yeah.
Look, forgive the slightly muddy question, but I'm trying to figure out what is the work that your discipline is trying to do. And there's a couple things that I heard, like when you drew the tree, there were not things connecting the tree, right? And when you talk about, I'm getting a little feedback. When you talk about the indicators of what might indicate things, it's staying away from saying like tool use is something that might have driven evolution. What is the question that this is trying to answer? Is it what drives the evolution of modern people? Or like, there seem to be a lot of very careful indisciplined things you're saying in very particular ways about how this all connects up. And what people are really incentivized to say that you're pushing back and saying, maybe we're going a little too far. But like, what is the work that people are trying to do here? Sorry for the muddy question.
Can you clarify? I can barely hear it again.
Okay. So you're asking like, what are the sort of big overarching questions that everything is feeding into ultimately? And yeah, I mean, it's hard. It's a good question. I'm trying to think of like how I would sort of define what those are at this stage in our discipline. I mean, there's so many of these like core, like big questions that have been around for like 200, 150 years at this point, like still have not been answered, because I feel like, I don't know, there was a point in time where it was like much easier to come up with lots and lots of questions without having to worry so much about like needing to be able to find an answer to those questions. Thinking about some of the big questions about like, you know, when, where, how, and why did we evolve to walk on 2 legs? Thinking about like what goes into that question, like there are pieces of that can absolutely never be answered. Like, you know, the why, what drove the evolution of this or this or this, like many of those questions are framed in ways that you could never know. Like, I don't know, we can come up with good guesses based on what the fossils look like at a particular time. But there are always going to be pieces that you can never know. I think in both of our cases, like for me, there's a big question right now thinking about locomotion, about what was sort of the variety, the spectrum of variation of locomotion that we see in the human fossil record. And so for years, there was this prevailing notion that, I don't know if you've seen the famous, like, they were from the old, what were they called? The Time Life books that sort of portrayed human evolution and showed there's like the March of Progress where you see like, yeah, the Descent of Man pictures, like, so those have found their way into popular culture and things of that nature too. So that classic image kind of made people think that locomotion evolved in this linear fashion, where it was like, every hominin was trying to get to modern humans, and like each one could only take a step that was so big, but incrementally they finally made it. And then like, we're the best, look at us, we walk in the best way imaginable. But you know, obviously that has changed, and that's kind of a silly way to think of this, but we didn't realize that until this fossil record really expanded, that we found lots more data to work with. Even in some of these time periods, like the late Tolly one, like it was still surprising to see that we have very different ways that hominins were moving at that particular time. And that diversity of adaptations is something that we're just beginning to appreciate, I think, as more and more pieces of evidence, pieces of this puzzle are starting to be found. I don't know if you would say something different for your particular area, but
Well, so hands, I've just spent the last half hour talking about how amazing our hands are. I mean, they are. But what I left off is that our skeletal morphology is so shockingly similar to that of chimpanzees and all of the fossil hominins. So yes, there are differences in robusticity and digit proportion and whatnot, but from the skeletons, from the osteological elements that are left behind, swap it out, like, it's so similar. So much of what we want to know doesn't fossilize, which is what Kevin ran up against as well. So a lot of what's going on is trying to understand how to tease out other information that we can't get from fossils themselves. What's going on with the soft tissue? And also, what is the extent of the variation that we see and the extent of the behavioral variation that is possible through anatomies that we didn't think were capable of such things. So I'm thinking of a few years ago, a researcher named Nate Dominy recorded chimpanzees engaging in a type of behavior that we thought was only capable of humans and gelada baboons for a particular reason. So they were engaging in this behavior, which is not that exciting until you it's realized that most primates can't do this in a forceful manner. But they recorded these videos of chimps engaging in really delicate precision manipulative behaviors, which suddenly made people say, oh, wait a minute, we have to reevaluate again, So trying to understand the spectrum of behavior that's possible given the morphologies that we see in extant organisms and the osteology of extinct organisms. I think that's a big question driving people in my area right now. I hope I couldn't quite hear your question. I hope I answered it.
My question is for Erin Marie. In your work with hominin fossils, examining them, I'm going to guess that the assumption is that most hominids were ambidextrous, but has there ever been any evidence, either from wear and tear or injuries or what have you, that some could have been, dominant, either right or left?
Yeah, evidence of handedness? No. We cannot, as far as I can remember from the literature, we can't even determine whether handedness exists in chimpanzees. And there are very few associated upper limbs. In order to try to figure out whether or not an individual was right or left-hand dominant, it comes down to a matter of comparative robusticity between the sides. But I am eager for the day when, we find these two arms just sitting in the sediment, but because it really exists on a comparative basis, we just don't have a sense. And of all of the stone tool makers that I've worked with, someone just asked when we gave a talk at Rutgers 2 or 3 weeks ago, whether there are differences in right-handed biomechanics versus left-handed when making and using stone tools. And again, I don't know, because we've had only one left-handed subject. So it's, as Kevin was describing with fossilized footprints, how rare those are, the fossilization of anything is really, really rare. And then to get associated fossils is just a dream come true, but it doesn't happen often enough for us to make any determinations, solid determinations about habitat.
Thank you.
Just to follow up on that, are there nappers who do things ambidextrously?
I'm sure there are, but I have not worked with them. That would be really interesting. I have not worked with them. And my research has gone in a really different direction. We're still analyzing some of this napping data. But my primary collaborator, Neil Roach, and I have not collected these data for almost a decade now. So we're still pulling information out of it, but I don't work with nappers that often anymore.
All right, we've got some more questions that had come in online. These are two anonymous questions that are going to be for Kevin that I'm going to just sort of combine together because they're very related. The first one, do higher foot arches improve the efficiency of human bipedalism? How bad are flat feet for this? And the second one is, what do you think of Lieberman's question mark, not sure if that's the right name, study that barefoot locomotion is superior to shoed locomotion, and how are footwear affecting foot evolution?
Yeah, so there's a lot there. So starting with arches, I don't think, I mean, it's hard to say absolutely that there's any difference in sort of efficiency between a high versus a low arch, because there are high and low arches. There are high arches that are very different from other high arches, and there are low arches that are very different from other low arches. And so it's kind of like there's lots of variation that you can have in foot anatomy, but you can arrive at sort of the same end result. Like you could arrive at the same efficiency with both a high arch and a low arch, or other people might differ with the same arch anatomy for other reasons. So I would say with that one, it's kind of too complicated to say for sure that you could sort of pinpoint and say there's an inefficiency difference between the two. Going to the sort of barefoot running versus wearing shoes while running, I think that was the second question, is that right? It's something where I think, there are advantages to going around barefoot. So our shoes, they do a lot for us. They do a lot of useful things for us. When we're walking around on concrete, when you're walking on a cobblestone street, they're going to protect your feet in ways that are really useful to you so you don't get injured. There are ways that walking barefoot is going to be helpful to you as well. We know from research that you can walk barefoot and improve the musculature within your feet. You can sort of build stronger, what we call intrinsic foot muscles, the muscles that exist entirely within your foot. So they're typically small. When you wear shoes, your shoes in a lot of ways sort of help support your foot. They help you push off against the ground in some ways. And so you can have a tendency to have smaller than ideal intrinsic foot muscles, and maybe going barefoot or walking or running barefoot or doing barefoot exercises might help you to build those muscles, and they could allow you to have a healthier foot. But I would say, again, it's sort of hard to speak in absolute terms. Of course, there are going to be situations where shoes are more beneficial. There are going to be other situations where going barefoot is more beneficial. And it's kind of like a matter of what are the trade-offs that you're going to be dealing with in those two situations. I remember when we were in graduate school in Washington, D.C., seeing someone running barefoot on the cobblestone streets in Georgetown and thinking that has got to be the most painful thing that you could possibly do. Go somewhere that's not a cobblestone street if you want to do that. But again, it's so variable where I think you couldn't say in absolute terms that one is better than the other in all situations.
We've got a couple more online questions here. And if anyone else has some in our in-house audience has some more questions, do feel free to flag Sammy down to bring that mic over to you. But if there are not many more here, then we're going to be wrapping up pretty soon. So let me see here. This is another anonymous one. I believe this one came in while Erin Marie was talking about, intelligence and, tool use and tool production. And the question is, does intelligence necessarily require bulk? I assume that means cranial bulk. Corvids use tools and their crania are quite small. So do
Right.
you have any reactions there?
There are, so on Darwin Day, or after Darwin Day at Duquesne, Duquesne always does a great Darwin Day talk. If you haven't seen it before, I really encourage you to check out their Darwin Day talk. But this year it was on, what was it called, of slime molds and
Slime molds and
swarms, and it was about how like slime molds don't have brains, they don't have neurons. And then swarming animals, it was talking about ants, specific species of ants. So neither species is something that we normally think of as being overly intelligent, and yet this researcher described really complex behaviors that they're each engaging in, particularly the ants. So I don't think that brain size is a great proxy for intelligence. I call the fossil, so I refer to the tyranny of the fossil record a lot. We are so limited by what fossilizes, and not only what fossilizes, but then can you find it? It's a big world. Can you safely get there? It's not a geopolitically stable world in a lot of places. So our store of knowledge is so biased by what we have been able to recover. So we're missing a lot of data, and the best, maybe not the best we can do, but one of the primary ways that we can, one of the primary proxies for cognitive capacity is cranial capacity right now. But it's absolutely there. If you have more at your fingertips, anyone who's working with extant organisms has so much more that they can say, and they would absolutely say it's not simply about brain size. There's so much more going on.
Great, thanks. And got one coming here from our in-studio audience.
Is it on? Oh, okay. I'm curious about the monkey with the giant stone to get one little nut. And I'm wondering why there's no evolution of tool making in that bunch. Why do they constantly resort to the same fatiguing, method of getting nourishment?
Yeah, good question. Why
No evolution?
So again, there's not a rich fossil record of capuchins. They're tiny. So another limit of the fossil record is that small things don't tend to be fossilized. They also live in hot tropical regions where, again, they're not going to fossilize. So we don't know a lot about or anything much about and the ancestors to capuchins. So it could be that that what we see right now is far more efficient than what they used to be doing. We just don't know. But they do gain a tremendous caloric benefit from nut cracking. They're not always lifting something that's 50% of their body mass. But even when they are, the configuration of their bones and their muscles and their tendons. And I don't think anyone has looked at this, but the fiber types, the muscle fiber types in their muscles, all of these things are working together to allow them to engage in this behavior that to me and you looks absolutely bonkers. They're engaging in it in a way that works for them. If it didn't work for them, they wouldn't be doing it. So they are adapted to lift those massive stones, 60% of their body length, and still extract some caloric benefit from it. Otherwise, they just wouldn't do it.
But then they never eat.
No, but I mean, they would find something else to eat. No species is going to persist in doing something that is not to their benefit. They'd either, as you said, die, they'd find something else to do. Yeah, they'd die or they'd find something else to do.
We need tools. That's the thing. I watched them, chipping this stone and I'm thinking, that's the key. That's a real tool. I mean, bringing a big stone over your head and cracking it down on a nut. Why don't you make it more efficient? Nutcracker.
I think what they're doing is amazing and it works for them. It works for them.
Fantastic. We'll do one or two more quick online questions here. And I always try to refrain from commenting on questions here, but I always like to think evolution's about good enough, not perfect. So
Yeah, you're right.
that's my perspective there. Anyhow, since I've got the microphone, but two other online questions here. This one is maybe a little bit, a little bit removed from your area of expertise, but we'll see if you have a reaction to it. And this could be for either of you. If locomotion is critical to an organism's survival, why can't plants locomote? So don't know if there's any thought there.
You don't know anything about plants. Why don't you answer that? What do you think it is?
Well, so I mean, you have to think about like why locomote, first of all, too. And so, locomotion has a lot to do with how you're engaging with your environment, your variety of social behaviors, your access to food, like how you're acquiring resources in your particular environment. And so thinking about the resources that a plant needs, like plants will grow in the places where those resources are available to them. And so given their sort of, capacities and lifespans and things of that nature, they don't necessarily need to move on a regular basis. Whereas organisms like us, we have to move all around our environments in order to get all of the food and all of the other resources that we're going to need to survive. So yeah, it's essential to us and it's essential to all of the other organisms we were talking about today in all those kinds of ways. Animals need to move to access the things they need, and not every animal moves necessarily, or not in the same way that we do. But it's all about how you're accessing all of those resources that you need to survive.
Also, plants do move.
Yeah.
So there are some that follow stimuli over the course of a day, but what I'm more thinking about is the fact that their gametes move, their seeds move. So the reason that fruit exists is to help plants' seeds disperse. The reason it's the same with pollen. Pollen and the gametes that exist within pollen granules are moved by pollinators. So maybe the tree doesn't move, but the reproductive components of trees will move.
Locomotion of a species, not an individual.
Right,
Yeah.
right.
Okay, great. Well, I think that's probably a good spot for us to wrap up for the evening here. And we've got folks online expressing their gratitude for you being here tonight and the lovely presentation. And I know our on-site audience loved that as well. We hope to see everyone back here with 2 or 3 friends or enemies for the next Café Sci. But get home safe, everyone, and thanks again for being here tonight. We appreciate it.
Sweet.